first of all, I'm Paul Brierly with Yuma Center of Excellence for Desert Agriculture. For those of you that don't know, we're a public-private partnership. We're funded by the ag industry, housed in the University of Arizona College of Agriculture and Life Sciences, funded by a lot of you in the room, and we appreciate that. So our aim is to work on problems that need to be solved and get them solved. So anyways, we got this center going about 2.5 years ago. Got our stakeholders together and asked what's one of the first things we ought to work on, and they said Fusarium wilt of lettuce. So we've been working on Fusarium wilt of lettuce. I'm going to give a quick overview of what we've done, and then we have Dr. Barry Pryor and Dr. Mike Matheron here to talk about some of the results and things that they've learned. The first thing we did was we asked Dr. Matheron to put together a 2-page analysis of what do we know, what is the disease, and what do we know, how can we work around it as best we can. That's this handout. It's at the back table if you haven't seen it. All this stuff is available on our website too, which is desertagsolutions.org. So hopefully that's easy to remember. But we appreciate it, Mike. We'd asked for a 2-page thing and it came out 3, but it's better than 25, right? Next thing we did was held an international symposium that a lot of you attended. We had researchers and industry from all over the world come and talk about it. That was this. I actually have a few of these left over. And on our website, we have all the presentations, the videos of the presentations, and their PowerPoints available. So again, if you want to see anything that was presented at that symposium, that's on our website. One of the things that came out of that was we had a brainstorming session the next day. And I know Matt led one of the sessions, but we had them on, we looked at cultural practices, containment and regulation, product development, breeding and resistance, and resources and funding. And kind of the bullet points of each of those topics are available back there as a handout and on the website also if you want to see what everybody kind of thought were the key things to move forward on. We also did a field trial at that point in conjunction with Dr. Matheron and Dr. Pryor to look at varieties, to look at products that try to control them. That was the first field trial that had been done in a long time. We did that in conjunction with that symposium. And those results and this year's field trial results are, I think, in your hands, if I'm correct. So hopefully those are handy. We'll hear more about that from Mike. And then we held a follow-up conference in Salinas, Monterey area last fall, basically to kind of keep things moving. And we focused on basically looking at resistant varieties and basically field trials and then research efforts too that we can move forward. And that's on a sheet that's also available in the back and on the website. We did the year 2 trials. You're going to hear about that. There's some ongoing research that Dr. Pryor is doing to be able to detect the disease. And we're also looking at working with UC Davis to be able to measure in the soil how much pressure there's going to be so you can help decide where to plant or not plant. And the other thing we're doing is working with USDA and a big data analytics company to look at what are the triggers of soil disease, and ultimately we want to come up with a decision tool that helps you make decisions that will avoid those triggers that cause the disease. You know, why does one field go down and the other one not? What are the factors that impact that? So in a nutshell, that's what we've done over the past couple of years, and I appreciate you all coming. We have our main collaborators from the U of A here, and I think at this point I'll introduce Dr. Barry Pryor to share with you what we know about the disease, what we've discovered, what we're working on for the future. Barry is a professor of plant pathology in the U of A School of Plant Sciences. And Barry, why don't you come on up?
So I've been tasked with sort of introducing again some of the findings we've had of Fusarium wilt, or the management of Fusarium wilt over the last few days or last few years, and I'm going to summarize some of the things that Paul has alluded to, things that are presented on the YCETA website, some of the things that are in your handouts included. I'm not going to talk about the variety trials or the product evaluations, I'm going to leave that to Mike. And I know a lot of you are here to listen about some of those really exciting findings. But I thought what I would do is sort of discuss some of the challenges, some of the insights, the updates, some of the advances, and to finish up talking about some of the new research that we're doing at the University of Arizona that Paul alluded to, some of our greenhouse studies for rapid detection. But I think it's important sometimes to periodically summarize our findings, because each time we hear it in a different context and in comparison with other, we start developing new ideas, we get new insights. And this is the key to managing Fusarium wilt of lettuce. It's not going to be a single solution, it's going to be a multiple solutions. And we're going to have to put together a program using a lot of different strategies and tactics to really address this problem successfully. So the symptoms, we're all very familiar with the symptoms of Fusarium wilt of lettuce. And of course, it's this vascular discoloration we see, this necrosis of the internal taproot, and it's a very distinct vascular discoloration. You cannot confuse this with very many other diseases. So once you pull up that lettuce plant and cut open that taproot, you can be certain that once you see this reddish-brown vascular discoloration, you're probably looking at Fusarium wilt of lettuce. Now, there can be a little bit of confusion. For example, sometimes we see, and I don't know if it shows real carefully, we see a black discoloration. Now, typically we don't see it as a black color, but a pathologist will start separating these two color symptoms. And if we see a black color, sometimes we start thinking of verticillium wilt of lettuce. We don't have verticillium wilt of lettuce here in the Yuma area or in the southern and southwest desert. Let's hope we don't get Fusarium wilt of lettuce down here. But in other areas of the world, Salinas, for example, they really have to look carefully at some of these symptoms to make sure they're talking about Fusarium wilt of lettuce or verticillium wilt of lettuce, and the management of these two are going to be different. And if we, for example, and again, I'm going to be showing some of the same slides that we presented at the International Symposium for the Management of Fusarium wilt of lettuce. So some of these slides are going to be redundant, and I hope I give proper credit to those who created these slides. But Steve Quakey, Extension Specialist, Farm Advisor of Monterey County, really put up a nice sort of table of some of the challenges is when we're looking at some of these soil-borne pathogens of lettuce. And as Steve says, if you're doing a drive-by diagnostics, and as you might say, look in the field and I see this disease and I see this disease, you might get confused with trying to diagnose Fusarium wilt of lettuce from some of these other fungal pathogens of lettuce that are also soil-borne, such as Pythium, Sclerotinia, and Phalaenopsis, verticillium. And you can see right here some of the symptoms: small stunted plants, wilted leaves, yellow leaves, collapsed plants. These are going to look very similar. And so you're really going to have to get out, walk over, pick up the plant, cut open the taproot, and look for that vascular discoloration to separate Fusarium and verticillium from these other pathogens, okay? So, so if this is really helpful if you're trying to look at a plant, you just won't be able to diagnose it just from that. And that's really a key right here, what I'm trying to say. Now, we've all seen the march of Fusarium wilt of lettuce around the world. It started in 1967, or a little bit earlier in Japan, and has since, you can see how it moved. 67 in Japan, 67, let's see, 93 in California, back and forth, 96 in Taiwan, 99 in Iran, 2001 in Arizona, and then we can see Italy, Portugal, Brazil, Argentina. It's marching around the world. And so the challenges that these people have with Fusarium wilt are shared by all of us. We're not alone here. We're not alone. And there's researchers in each one of these areas that are also looking to manage this disease as well. But then we have to start, and in fact, I think there's a report now in Netherlands in 2015 or 16. So it's still marching around. And we have to say, how did it do this? How did it move all over the world? Did it independently arise? And of course, one of the primary things we think about is infested seed, transmission via contaminated seed. Now, this is a common mode of transmission of Fusarium wilt in other crops. There's a lot of different kinds of Fusarium wilt. Fusarium wilt of, say, tobacco or melons or cotton. And these are also can be seed-borne. So we know that Fusarium wilt pathogens can be seed-borne. So we said, well, is that a problem here? Is that a problem here? Well, some of our early work of Fusarium wilt was to look at some of the seeds that we're using here in the Yuma area. And we used, in this study here, we looked at both raw seed and pelleted seed, and we looked at two different methods: direct plating, where you're actually going to put the seeds on a petri dish, and a DNA-based method, a PCR-based method, where we could look at the signature DNA of these pathogens. And over a 2-year period, we looked at 88 seed lots. Growers sent us in seed lots, seed companies sent us in, and we found none. We found none. Even though we could spike these seeds with the pathogen and recover it, so we knew our methods were good, we found no contamination in the seed. And so we said, gosh, can it be seedborne? Can it be seedborne? So we did some studies, of course, in the greenhouse, where we looked at, planted some tolerant lettuce varieties, and we wanted to see, can we infect these lettuce varieties and see the pathogen move up into the seed? And of course, we had to use tolerant or resistant varieties because we had to make sure they survived, right? You didn't want to infect a plant and have it die and not get any seeds. So we had to use as tolerant varieties as we could, and using romaine varieties and things like that. And we grew plants to maturity, we harvested the seed, we assayed for Fusarium oxysporum lactococcus, all from the roots, all up the inflorescence and up into the seed. And what we found is that as you move up into the inflorescence, the recovery got less and less. The pathogen just did not move up there very easily. And then we also found that the seeds did not become internally contaminated, whereas the inflorescence would become internally contaminated. As you moved further and further up, you got less and less of the pathogen, and the seeds had no internal contamination. However, if we took these seeds and we mixed them with infested debris, they became, like during the threshing process, they became infected very easily, very easily. So this was the source of the contamination. Not that it was internally born, that it was externally born. So we start thinking about this as means of managing this disease. Now, the researchers in Italy were doing the same sort of thing, and they had different results. They had different results because they looked at 27 commercial seed lots, and they found 9 of them were infected. Now they targeted seed lots that were grown in infested fields. So they were sort of, how do you say, they were sort of fixing their methods so that they would find that pathogen. And they did, but they also find that the seed contamination occurs at the seed surface, not internally. And they also found, of course, we found the same thing, all of these seeds were race 1. We've heard about race 1, race 2, and race 3, and all around the world we only have race 1, and in Japan they have race 1, 2, and 3. Now, of course, management of soil-borne inoculum, we're familiar with this. We're familiar with managing soil-borne inoculum on a number of different vegetable crops, and there's standard ways we treat this with hot water or chlorine, hot, dry air, different fungicides, whether they're conventional or organic or biological. And then, of course, we have to consider all of these other parameters. And, of course, the seed producers know very much about the importance of managing these parameters to maintain the quality of those treated seeds. But I'm not going to go into seed management, but there are tools. There are tools we can use for managing seed-borne inoculum. But, of course, one of the other questions is, how about transmission via infected soil. We know that the soil become infected. What happens when a tractor or workers move through a field and then come out of the field and there's mud and they're carrying soil with them? There's many Fusarium species we know very well that move through contaminated soil. It's a common mode of transmission. Okay? And the proximity between Arizona and California sort of suggests this might in fact have happened because we know the movement of equipment and different kinds of material used in lettuce harvesting and lettuce production moves from Salinas to Huron to Yuma and Imperial and back to Huron and back to, and we know that there's this cycle. We know that the equipment moves. We suspect that there's also soil move. But you know the interesting thing is we've not yet done a systematic study of looking at soil adhering to the farm equipment that's moving in from California to Arizona. We haven't done those studies. We assume it happens, and we make an assumption, but we haven't done the studies. What we have done, though, is we've looked at genotypes in the soil in Arizona and compared them with genotypes in the soil in California. Now, this is a little bit different than races. It's a little bit different than races. Think of races almost like separate species, not really species, but they're quite distinct. Within each race, there are genetic fingerprints, signatures of strains that you can follow. and that are characteristic of that strain. And we can find the genetic signatures of all these different strains in the Yuma area. And then Tom Gordon over in California in his research program also looked at the genetic signatures of strains in California and found that the genotypes in Arizona matched that of California. So we really kind of know that what happened in California came to Arizona. Did it come by seed? Did it come by soil? We're not quite sure. But what's interesting here is that this was down in 2001, 2005. We haven't done these studies in a long time. Perhaps it's time to readdress this. Perhaps it's time to go back and look at these genotypes, okay? And you know what's also interesting is we haven't done that on a worldwide scale either. You know, we've done that between Arizona and California here, but have we done it between Japan and Taiwan? Have we done it between Brazil and Argentina? Now, although we imagine that the seed transported the pathogen around this area, was the seed responsible for those local transmissions in neighboring countries? Or could that have been through infected or infested soil? So, management of soil-borne inoculum. This is also very important. And you can see the importance here because when we have 100 to 150 propagules per gram of soil, we can see the damage this does right here. Now this is not a high population, as you're going to see. This is not a high population. And we can see the damage that occurs with just these lower populations. So we've looked, or researchers both in Arizona and California have looked at ways to manage soil-borne inoculum. Mike Matheron has done a number of studies here, and this is some of his work he presented earlier on solarization. Solarization, we know that solarization works for other soil-borne pathogens as well. Solarization performed during July and August when it's really baking down. And then look at the results here. Plots planted to susceptible crisp head cultivars. Pretty good results, okay? And these are the years Mike has done this study, 2004 to 2007. Look at the percent reduction. 42% reduction, 81, 98% reduction in disease. This is an effective method. This is an effective method in perhaps soils that would not enable you to produce a crop. Whether it's economical, those are decisions that field managers and producers need to make. But the fact is, it does show nice efficacy as well. So we've also looked at dry fallow to reduce soil inoculum levels. And this is just leaving the field dry and literally abandoning it for a while. And these studies have been done by both Mike Matheron and Tom Gordon in California together in conjunction with each other. And the graph I'm showing here is some of Tom's work on some of these micro field plots, just leaving them dry. And it's important to leave them dry and not planted, and you'll see the importance of that in a minute. But you can see how the colony forming units per gram of soil have gone from 3 to 4,000. It's pretty, remember the other slide I showed you, 100 to 150 propagules. This is 3 to 4,000, really heavily contaminated soil. And look how long it takes to come down to 17. And you know, so right up in here, we're still at 15 months, we're still at, you know, several hundred in here, still for a long time, a long time. However, this method can work. How you guys fit this in, how producers fit this into their management strategies, it's another tool. It's another tool. And this is what I'm trying to present here. There's a lot of different tools, and we've got to perhaps think about them. Different fields are going to require different tools. Different areas are going to require different tools, right? Okay, crop rotation. Now this is what I want to get to as well. Some people, we think that maybe if we move to different crops that aren't symptom hosts for Fusarium wilt of lettuce, we might be able to perhaps deplete the soil of the pathogen, you know, sort of wait it out in a way. And here, this is some work that Tom Gordon had done in California, looking at rotations, or actually planting different crops in infested soil and then seeing if he could recover the fungus within the plant tissue. And these are colony-forming units per gram of cortex, of root cortex. So he was taking the roots and he was trying to isolate the pathogen from the roots, cauliflower, broccoli, and spinach, and this is the amount he was able to recover. And as you can imagine, lettuce, of course. He was able to recover the pathogen from the lettuce cortex. But look, he's also able to recover the pathogen from spinach, broccoli, and cauliflower as well. from the cortex tissue. He looked a little bit further and inside the vascular tissue, because this is really where the fungus moves up and down inside the plant. And again, look at those 4 plants. Lettuce, of course, there's a lot of the pathogen up inside the vascular system of the lettuce plant. And spinach also, it's up in there. It's reproducing in spinach. It's proliferating in spinach, although spinach isn't showing symptoms. But then broccoli, there's a little bit, and cauliflower, he really couldn't find it much inside, so it doesn't colonize cauliflower very well, all right? So what we learned from this is that Fusarium wilt of lettuce will colonize the inside tissue of these other plants that are not hosts, that are not symptoms hosts. And it'll even colonize resistant cultivars as well. And we call these cryptic infections. You don't think that the pathogen's there. It's not causing the disease, but it's there. It's there, and it's reproducing, and inoculum is increasing, and then when you move this infected debris back into the soil, you're moving the propagules back into the soil. So what we learned here is that some things are not good rotational crops. Some crops, alternative crops, are not good in a rotational strategy. And we got to even remember that when we're planting resistant cultivars and moving that tissue back into the soil, we're probably increasing soil inoculum as well, even though you're not seeing disease, okay? But there are some other crops that might be less efficient hosts. Cauliflower and broccoli might be colonized to a lesser extent. And so if we are another tool thinking about crop rotation, we have to think very carefully about the crops we're going to use. And so these studies, we show that cauliflower and broccoli. What about other crops? What about other crops? What about grain rotations? What about other kinds of vegetable rotations? Summer rotation, melon rotations. Maybe these studies need to be done. So I hope that what we're bringing home from here is that, you know, we're sort of summarizing some of the studies that have been done, but also pointing out some of the gaps we still need to fill in. There's a lot of information we still need. Okay, and of course this was one of the really a key part in the management, how to manage the wilt, the disease, in the presence of infested soil. And these studies have been done, of course, and you're all very familiar with this, right? The specialization of this pathogen for warm temperatures. And the expression of disease is much higher in warmer temperatures, right, when you're planting in September than when you're planting, of course. And this is a really important management strategy. I'm sure you all are using this regularly, okay? So, and this was some of the work that Mike had presented already, and very important work, very important. Again, these are all different kinds of strategies that we can use, that we can employ. Hopefully in combination. Okay, so what I want to finish up with is talk a little bit about some of the research we're doing to complement all these other strategies, right? We're trying to complement them. And one of the things we want to do, and I should probably, no, I won't go back all the way, but remember, we saw that Fusarium taproot, that infected Fusarium taproot. The internal tissue is all necrotic. That lettuce plant is gone. There's no production from that lettuce plant. What would be really nice is if we could detect the infection early on, as early as possible. And maybe there might be other management strategies we can bring in on top of that infected lettuce plant to make sure that infection doesn't go any further. So even though that lettuce plant becomes infected and might be susceptible, it might act like a resistant cultivar if we get perhaps the right product on there, maybe a plant defense enhancing product, apply it. So we're trying to look at detecting the pathogen as early as possible. And we're going to do this in a couple, and these are some of the objectives for our 2016-17 Specialty Block Grant Award from the Arizona Department of Agriculture. We have 3 objectives. We're going to look at rapid DNA detection by isothermal PCR. It's a new type of DNA-based method of detection. And then LAMP isothermal PCR, which is loop amplified, no, Mike, help me out here. Loop amplified isothermal amplification. I can't remember what the acronym stands for, but it's a modification of this isothermal PCR amplification. Really, they're focused on field diagnostics, tools the grower can use out in the field. And then we're going to also do something very cool. We want to use you've heard about this as well, multispectral analysis of plant stress using very fancy cameras, right? To see if we could identify plant stress, whatever it is, early on, as early as possible. That's the key, right? And then, of course, part of this is going to be chemical and biological product evaluations that Mike's going to talk about, and I'm sure you're all very interested in seeing those data as well. So our objectives 1 or 2 are greenhouse-based studies. These are greenhouse-based studies where we set up a large greenhouse, fill it with lettuce plants, use a randomized complete block design, and we're going to stress these lettuce plants in a number of different ways. We're going to stress them with water, right? We're going to do 2 different water levels, stress them at 2 different water levels. We're going to stress them with 2 different fusarium inoculum levels. And we're going to compare that with another pathogen you're all very familiar with, 2 different levels of Sclerotinia sclerotiorum. Actually, in this one, it's Sclerotinia minor. And then, of course, control is no water or biotic stress. And after we stress these plants, we're going to go in and on a weekly basis, we're going to sample these plants destructively. Of course, we have to remove the plant. We're going to remove plant tissue, isolate or analyze it for those signature DNA of Fusarium Oxysporum lactucase, the Fusarium wilt pathogen. And then we're going to also look at these plants using a multispectrum camera to identify stress. And what we want to do is we want to see, can we differentiate stress from water or pathogens? Maybe is Fusarium stress going to be different than Sclerotinia stress? We don't know that. These studies have not been done. But the objective is to see how soon we can see if that plant is stressed and perhaps what's causing the stress. And so here's our fancy Mica Sense Red Edge camera, right? It's actually a very cool, it's a small camera. You could probably put this camera on a drone. I think you can put it on a drone, fly it over fields, and that's sort of our long-term vision, right? You guys have heard these discussions as well, putting these cameras on drones. And it actually has 5 different cameras in there. And the 5 different cameras capture 5 different spectral bands targeted at agricultural applications and really targeted at points of plant stress. And you can see these 5 different bands here, and we can see blue, green, red, near red edge, near infrared, and we can see the difference in this green line, which is a healthy plant, and the difference in spectral response versus a stress plant. So we know that these plants are going to respond differently. And this camera should be allowed to see very sensitively these stress signatures. What we're hoping to see is these stress signatures are going to differ between a biotic stress and the kind of biotic stress and maybe the water stress and maybe different kinds of abiotic stresses. Maybe salt stress would even give a different signature. So these are kind of some of the things we're looking at. And if we could see this in the greenhouse, of course, Paul's vision always is put it on a drone and move it out to the field, move it out to field applications, okay? And here's our imaging system right in here. This is our little imaging box, and it's very light, tight, and we fix the camera up inside there. We put 3 plants in there at a time, and we measure their spectral images, like I said, 3 times a week. Here's how we perform the water stress, a very sophisticated, oh, and I should say, actually, I should back up, because this is in collaboration with Dr. Murat Kassira at in the Department of Agriculture and Biosystems Engineering at at University of Arizona in Tucson. And he is sort of the engineer behind some of this experimental setup, particularly the water stress. He's a very fancy injection system. And Murat is very sophisticated, very knowledgeable in how to create and hopefully respond to and sense water stress and salt stress and abiotic stresses in plants. And then, of course, we have our biotic stresses. We've included Fusarium and Sclerotinian in here. And in fact, right now, these experiments are in progress right now. And we can see that we have plants dying. You know, plant pathologists love this. We love to see our plants die because we know we did it, right? And we see right in here, you see this wilted leaf and this necrotic vascular system right there. But interesting, the plants that are dying right now, they're not dying due to Fusarium, they're dying due to Sclerotinia. Ugh, that one just melts plants fast. That's an age-old problem that you're all very familiar with. But it's really important that we compared Fusarium with Sclerotinia in this experiment. Okay, these first sets of experiments are currently underway. We're going to continue these experiments for another few months, so stay tuned, okay? So sort of in summary, management summary. Again, put it all together. This is a problem that's going to require several tools, many different kinds of strategies using pathogen-free seeds, starting with That's first and foremost. Every vegetable grower knows this. Know the history of the field that you're going to be planting into so that you can anticipate what kinds of challenges you might have and be prepared for those challenges using the most resistant varieties available. And Mike's going to be talking about that. You avoid susceptible varieties in warm winter plant. These are things you guys already know, but we need to bring them together in an integrated management strategy. Remember that soil inoculum, this is important for the future. Soil inoculum can increase on all varieties, whether they're going to show symptoms or not, and other non-lettuce crops. Many non-lettuce crops, okay? Broccoli and cauliflower seems to be the better rotational crops. Better, but not the best, perhaps. More studies are necessary here. And that inoculum can be moved with soil. So, of course, good sanitation practices are critical for managing fusarium wilt of lettuce, as with all soil-borne diseases. All right, and I think that's it. Paul, am I taking questions, or do we wait for questions at the end?
About 5 minutes.
Okay, so thank you. I'll be willing to be happy to answer any questions you might have.
Could you go back to that slide where you did the solarization, please?
And now I should probably ask Mike to answer questions on this one. This is Mike's work, and
Is that the same field? The left photo and the right photo?
Mike, it's the same field, a different perspective, close up, far away.
One's 180 degrees, the opposite,
Oh.
but it is the same field.
Turned it around,
Yeah.
right? But you can see we have solarized, non-solarized, non-solarized, alternate. And look at this, nice, diseased, healthy, diseased. You know, I don't know. Mike has the what's this?
Leave that plastic on that field.
Mike, how long?
1 month,
1 month. And Mike was
When did you do it? What time of year?
summer.
during July and August.
Yeah.
Why can't you do that with water? Why doesn't it
Mike?
work with water?
Mike, does it work with water?
It doesn't get hot enough, probably. No, it does not work with flooding.
Mike says it does not work with flooding as well. Probably doesn't get hot enough. Probably doesn't get hot enough. And Mike, were you able to establish decreases? Were you sampling the soil at the same time during this study? Okay, so we don't know what the decrease or lack of decrease in soil populations. In this study, we're looking at the end results, which was harvestable heads.
I can't read that. What was the end result?
The end result, 2004, 42% reduction. 2005, 81% reduction. 2006, 98% reduction. And 2007, 67% reduction.
What's that cost per acre for that plastic?
That I can't answer. I don't know. Maybe some folks in the audience would know that answer for cost per acre in putting down plastic.
Would it be better to use black plastic?
No.
It probably wouldn't, because we need the sun to, we need solar penetration down into the soil.
It's more the solar than the heat, but is there, what is your moisture level when you put your plastic on? Have you,
Well, basically, the field was irrigated.
Okay.
And then as soon as we could physically go in there, we laid the plastic. So you need moist soil
you
as
need
well.
moisture, but you don't need,
Right, dry soil,
Full
doesn't,
soil,
it's not
yeah.
as effective.
Full profile,
The
okay.
wetter soil, the better.
yeah.
Yeah, hold on, Mike. Yeah.
With regards to water, he mentioned it wasn't really the heat, it's more, solarization, the sun.
Well, it's a combination. The clear plastic allows the solar penetration, that the heat travels deeper into the soil. And what the plastic does, basically, it retards the loss of heat during the evening. Whereas if you're flooding, that's another thing. The flooding, the water is able to take up a lot of heat itself. So the water does get warmer, but that heat does not penetrate deep into the soil. And now, we haven't tested all those parameters, but basically we have, when we have flooded soils, the temperatures don't rise as high in the first 6 or 8 inches of the soil profile compared to if it's solarized. This type of solarization, you could get temperatures up to 150, 155 degrees. It peaks out there, it doesn't stay there, but every day it goes up and down, and that tends to kill the fusarium.
And it kills many of the seeds as well, weed seeds as well.
Right.
Up at
It
that
is
temperature,
good for that as
nematodes as well. So you're doing a pretty effective kill of a lot of the biotic challenges you're going to have in your field. Yes?
Why do you think that was so inconsistent on the results, Mike?
First year, that was this trial. We were basically, the first time we did it, we weren't very efficient in how we did the trial. We got progressively better, and as I jokingly say, I should have stopped after 3 years. Fourth year, again, I can't remember exactly what, we had some technical problems there as well. Where this would probably really work better if you had a drip irrigation system where you could supply moisture while the solarization process occurs. That way you keep the soil moist, not saturated, but moist, and that helps the solarization process.
And this is sort of shows the challenges with doing field research, because we like to try to control all of the parameters. It's pretty dang hard to control them all. So we have to repeat things again. We have to do it again. We have to do it again. And then over time, we're going to hopefully sort out some of those variables and understand the varying parameters that are going to either result in 42% or 98%.
Mike, were they done in the same location every year, or was it different locations?
They were done in the same field, but not the same location. You know, we did use the virgin place each time. Yeah.
So that might also affect if you have different populations of soil. Maybe if you have high populations in one area of the field, you might not see a significant of a drop, or lower populations in another, you might see a lower. So again, multiple locations, multiple years. It's key for understanding field trial results.
If you knew you had both scleral and fusarium there, what would you do first?
I'm going to leave that one to Mike. He knows this much better.
Which one do you want to get rid of? Because, as you know, the flooding is very good for knocking out Sclerotinia sclerotia, but fusarium is not. This technique will help rid sclerotia as well, and weeds, as Barry mentioned. So perhaps you want to do this first.
What do you think the soil depth is in the solarization that you did?
Pardon?
The soil depth, how deep?
How far is it effective? Well, we did, I don't have the slides of this here with me, but obviously the first 2 inches, it's very effective. The soil temperature is very high. It drops down. When you get to about 8 inches or so, you're not really getting very much heating.
Do you work those beds up again, or do you mulch it, or do something?
Well, basically just lightly mulched it and planted after that. Now, if you solarize and then decide you're going to, if you flat solarize and then shape your beds, you're going to be defeating the purpose, because if you're digging soil below 8 inches and forming beds, that's not a good thing to do.
Yes, in the back.
Can you go back to the fallowing slide?
Oh, right. This one. Yes. Is this one?
When do you reach zero?
Oh gosh, well it hasn't reached zero at 35 months. It has not reached zero. So this is going to be one of those kind of graphs where it tapers off less and less and less and less and less and less for a very long time. So maybe if you follow this out for maybe 4 years, you still might come up with a few propagules. It might be really hard to reach zero, but that's okay. 3 propagules per gram might be okay. You know, so we're looking for what's the end result. End result is we want a crop. At which level do we get the crop? Do we get a crop with minimal loss? 0, of course, is good, but maybe 5 is also good, or 10 is also good. Those kinds of studies are also very important and maybe haven't been done to the completion that we'd like to help you guys in terms of management.
Do we know the soil
Kevin.
temperature that actually kills the pathogens?
Well, I think people have worked out studies with solarization fairly well. I'm not an expert on solarization. Mike's much more so because he's used it a lot more. But what do you say? 160 is pretty dang hot and kills a lot of stuff. Usually when I teach plant pathology, we like to get, I think 152 is sort of a magical number where you're going to be killing a lot of pathogens, a lot of seeds. Now some things are going to survive past 52 degrees C, I'm sorry, 50 degrees. Some of these pathogens are going to be surviving past that, but you're going to knock out things pretty heavy at those kinds of temperatures. So I'm not sure what temperature you're going to get. Gosh, do you want to kill everything? Probably not. That's a big area of research right now. This whole soil microbiome, right? The soil community. How does that impact Fusarium wilt of lettuce. Those are studies that need to be done more to completion, because if you knock out everything and you have sterile soil, you know, there's an axiom, a statement by even Aristotle. He said it: nature abhors a vacuum. Means if you wipe out everything, some bad thing's going to come in and take over. So you just don't want that. You want to keep a community there. You just want to knock out the pathogens and try and leave the community, most of the community, intact. People are understanding the importance of that community en route health more and more, and we're learning that sterilization is not necessarily your objective. It's not necessarily the best thing. Yes.
And in our desert soils, fallowing just brings your salts to the surface, and then you're fighting additional problems.
So there's
The minute you go back.
Yes, there's another comment about how fallowing is going to impact salinity of the soil, and so those are also problems. You know, Mike Stangolini, who was at U of A for a number of years, did some really interesting studies on irrigation and controlling Monosparassicus cannonbalus, right, in melons. And he found that if you maintain, when you did this furrow irrigation, you had these periods of flooding and dry and flooding and dry, that just exacerbated the pathogen population. You know, because as soon as you flooded it, okay, the community would go up, but when you dried up, the community would go down. And the first bit of moisture that came in, because you kind of have a vacuum there, sort of, the first bit of moisture that came into that soil, the Monosparassicus cannonbalus would just take off and have big problem. But if you maintained a little bit of residual moisture in that soil, your incidence of disease was less. And it was probably because that whole microbial community was intact and helping to perhaps repress a little bit the impact of those pathogens. So again, solarization, we have to remember our target, not to kill everything, but to kill the bad things.
So I'll continue basically talking about the work that Barry and I were involved in the last 2 years, looking at cultivars and also crop protection products for hopefully finding something that will help us reduce the incidence and severity of Fusarium wilt. All right, this is the bad guy. This is Fusarium. You never see any of this. The mycelium that you see in the upper left, that grows on a petri dish, and we can actually visualize it then. But in the plant, this is all growing internally, and so it would be invisible to us, as well as the spores. You're not going to be seeing those because they're microscopic. But what they do to the plant, we can actually visualize that with our eyes. And these are the symptoms. Barry has showed you this already. And as you know, those of you who have had to deal with this disease, you can get a lot of these symptoms. You can have patches within a field where the plants are basically all destroyed, and sometimes the entire field can look like this. So, as you know, I don't have to tell you, it could be very devastating disease. So we're going to be talking about lettuce Fusarium wilt. That's the Fusarium oxysporum that only attacks lettuce. But there's all kinds of other types of plants that succumb to Fusarium wilt also. And for almost all of those diseases, the primary tool that's used is to try to find genetic resistance to the disease in whatever plant you're talking about. And lettuce is no exception. So that's why we look at cultivar evaluation as a potential tool and perhaps the primary tool that could be used to manage the disease. So in 2015, we delved into this for the first time, other than the initial looking at cultivars in the early 2000s. And we looked at the crisp head varieties you see there on the left, as well as, interesting, okay, things changed a little bit. The romaine varieties that were looked at, examined this time. You can see there we had 2 growers' fields, both located in Yuma, and how the plots were established and so forth is described there. Basically, we had 4 plots, each 100 feet long, randomized within this field so that we could do the proper statistical analysis of the data we hope to achieve. And we planted these early to mid-September, and they were both harvested in mid-November. Again, we chose specifically the time of the year when the disease tends to be worst, if you will. Again, the numbers you're going to see as far as what we
how we rated the plots was looking at the percent dead or diseased plants. And so obviously you see in this picture there's some dead plants and diseased plants. And we before that we would test some of these, so we know that these all died due to Fusarium. And so the numbers you're going to see will reflect these numbers. Okay, so we had 2 trials, and we'll take trial number 1 first. You can see the lowest incidence of percent dead or diseased plants was 2, going all the way down to 31. Now this data is all in your handout, so we'll go through this relatively quickly. But you can see where the least amount of disease occurred happened to be in the romaine varieties. And this is actually what we found early in the 2000s as well. Romaines in general, and certain romaines in particular, were very resistant compared to their crisphead cousins. And so all of the other varieties that are listed there, you see had higher and higher levels of dead and diseased plants. This does not mean that everything else was marketable, because we did not use that particular metric in these trials. The second trial, again, you can see overall the numbers tend to be smaller. The amount of disease here was less in this particular field. As Barry mentioned, the amount of disease that you get is related to the amount of the pathogen in the soil. And we actually did do an analysis, and there was less of the organism in the right-hand field than the left. But you see the same result. The romaine varieties did the best with respect to less of them dying or becoming diseased. And the crisphead varieties that were tested here, unfortunately, didn't do as well. And this is how it looked in a graphical sense, and you can easily see here that there was a lot more difference in one field compared to the other with respect to the amount of disease. But the amount of disease in particular varieties held up irregardless of that initial disease level. All right, the next graph I'm going to show you is data presented the same way, but for 2016, or last year. Quite a bit different. Now this time we had a field from Yuma. Actually that field was one of the fields that we had in 2015. And then we had a new field, one in Welton. And you can see that the Welton field, signified by W up here, we had a very hot field with respect to disease. Very, very high level of disease. On the other hand, you see the 2 romaine varieties up the top, Del Sol and Duquesne, very low levels of disease. And what was encouraging to us was that 2 cultivars there, Oracle and Meridian, are crisphead varieties. And they were much less affected by disease compared to all the other crisphead varieties that we had here. So that's encouraging. Now, those were the varieties that were in both trials. We had some varieties that were in one trial and some the other, so that's why I'm showing this particular graph here. So again, this is the Yuma trial, where the disease tended to be less severe, and we had disease from as low as 2% dead or diseased by the metric that we used, all the way up to 43% in the varieties here. And we only had 2 romaine entries in here, and again, they were both right up the top, least amount of disease. And then there's Oracle, and Meridian is down a bit lower. The 2 crisphead varieties that did fairly well, or quite well actually, compared to any other lettuce, crisphead lettuce variety. And then there's some other numbered varieties there. Those are some additional crisphead genetics that were in this trial. Unfortunately, they were not in the trial where we had the real high disease pressure in 2016. But here, they did very well compared to some of the other crisphead varieties that were tested. In Welton, where the disease pressure was very, very high, basically anything that was a crisphead variety other than Meridian and Oracle, was basically a complete loss, 95% to 99%. Meridian and Oracle, crisphead varieties also, the newer ones in development. In comparison, we had 8 and 13% loss. Again, I know you'd all like to see 0, but to me, this is a great step forward. And of course, the romaine varieties, there were actually 4 in this trial, had very low level of disease. Okay, picture kind of tells it all. This is the Welton trial. If you can visually eliminate the weeds that are there, there's not much green. But in the background, you can see there is some lettuce that is actually alive. And if we look at those 4 plots a little more closer, they are the 2 varieties of romaine that were in this sequence of 4 plots. And then the 2 varieties, this should be Oracle over here, it was cut off. The 2 crisphead varieties. So anyway, this to me is very encouraging. We're on the way. At least we have now 2 cultivars that are basically somewhat resistant to the disease, very resistant compared to any other crisphead variety we've had to date. Now, some growers, obviously many growers have looked at these and maybe they're not quite ready for prime time yet, but me as a plant pathologist, at least they're alive. And so from further working on these, perhaps we can get them to the state where they will be commercially acceptable during the time of the year when this disease is most rampant. Okay, the other part of the research I will discuss is the crop protection products. And again, here the goal is to find, our goals are somewhat limited. What we're trying to find is something that will reduce the amount of disease. Again, for those of you who deal with some other diseases like perhaps powdery mildew or downy mildew, where you can achieve a high level of disease control, our goals aren't quite that lofty because with Fusarium, nobody to this date has really found anything that will basically knock it out. So we're trying to find something that will help give us a tool that, used in conjunction with other tools, as Barry mentioned, when put together, can give us a useful package that can be used to manage Fusarium wilt. So basically we used the variety Raider here because we knew from previous research it was one of the many crisphead varieties that is susceptible to Fusarium wilt. The first application of these products that were, again, these trials were in both trials in 2015 as well as both trials in 2016. First application was always made at seeding and right before the sprinkler irrigation water was turned on to germinate the seed. And the material was applied in a 4-inch band with a CO2 assisted backpack sprayer. And then most treatments were applied again right after seed germination. These are the products that we looked at in 2015. We have Actigard that you may be familiar with. It's basically it's a plant resistance activator. The other products here are all biologically based in nature, as you can see here. Either question?
These were just sprayed on top of the bed, is that right?
Sprayed right on top of the bed, and then the sprinkler irrigation. The idea was that the sprinkler irrigation would move them deeper into the seed zone. But they were sprayed.
Have you done anything where it was injected into the soil and the seed line like a dryer or something like that?
Not as of yet. No. No. Our theory here is that, especially in highly infested fields, the plant becomes infected basically as soon as the little radical comes out. So if you were to inject, I'm not saying that wouldn't work, but it would have to be there right when that little young root comes out, because as soon as it comes out, infection probably can occur very quickly after that, because plants actually start dying even before thinning occurs. So the plants are very small when they actually become infected. So that was the rationale for doing this. But that is something we could look at in the future. So these are the products that were examined at the time, the active ingredients on the right. And this is basically what we found. Now, the numbers here are different. This is the number of healthy plants in each plot. We've been talking about the number of dead plants or diseased plants before. So you see the non-inoculated control over here, it's underlined. That was basically 48 plants were healthy. And the best treatment, if you will, was Root Shield Plus. You see up there with 58. So we did get an increase in the number of healthy plants, which is something we're looking for. But statistically, you see the A's and the B's that are there. The only product that was statistically significant here was the Root Shield Plus in this data for 2015. So that's good. That's encouraging. We had something that actually looked like it could be promising. The other ones, even though they're not statistically significant, you've probably heard me say this a lot, one trial really doesn't tell you, if you see something that's good, that's good. Hopefully we can repeat it next year. Even something that maybe in this trial didn't look too great, we still trial again to see if we get the same result the second year. So this is basically how all of these treatments sorted out. 2016, we got a little more energetic. There's a lot more treatments on this list. We actually, the top 4 materials are actually, well, as you can see there, the first product, it's a conventional fungicide in development by Syngenta. Then there's a prothioconazole or Proline. Mancozeb or Manzate, that's been around a long time, but previous research from that International Fusarium Conference showed that perhaps this had some activity, so we included that, as well as Actigard, which we had tested the year before, plus everything else here are again biologically based materials. And we're not done. There's a few other products here as well. So all of these were put in, randomized in the plot, and these are the numbers we got. Now, in Yuma, in 2016, the Yuma trial where the disease intensity was less severe than it was in Welton. So this is the breakdown. You can see here the average of 13.5. We're going back to dead plants now. 13.5 dead plants with no treatment at all compared to 8.5 with the Tegro. But you don't see any letters up there. Unfortunately for us, because of variation between the replicates, there was no statistical significance between these. So we can't really say much about this. Numerically, they're listed from highest, well, lowest to highest, highest to lowest, however you want to look at it. But because of the tests that we routinely use to determine whether this is a real thing or not, we could not find anything here in this trial. Welton had the number of dead plants or diseased plants obviously is a lot higher. And again, the non-treated here was 145. The highest numerical increase, if you will, highest in that it's lower in the amount of dead or diseased plants, was 132. But again, because of the variation within here, statistically, we can't make any conclusions from this. So, bottom line, from the crop protection standpoint, we really haven't found anything that's a standout, stellar performer. Doesn't mean we're going to stop. We're going to continue to try to look at these. And part of it, what perhaps we can improve on is what was suggested, different delivery methods perhaps will improve. Because each of these products is unique unto itself. It has its own characteristics. And applying them all the same way is perhaps not the best way to go. So again, this basically is what the crop protection products trial looked like, again, in Welton. Welton was a tough deal. I mean, again, it just shows you those 2 crisp head varieties that basically we had some living tissue there. That's quite a remarkable achievement. So where do we go from here? These are some of the things for the field trials that we're thinking of doing this coming fall. We want to continue the cultivar evaluation, not just the 2 or the other ones that we found this year, but any others that might be of interest and want to be included by seed companies. We haven't given up on the crop protection aspect. We want to look at anything that shows some promise, something new, perhaps that we didn't test before, or going back to what we did test before, finding a different delivery method or something that'll help perhaps enhance the activity or that we found. Cultural practices. We're going to be looking at some of these in cooperation with the grower here, looking at fallow, deep plowing. The idea there is perhaps take the highest level of inoculum, which tends to be on the soil surface, inverting that so it's down lower to see if that has any effect on the subsequent lettuce crop that's planted. Seed treatments. I forgot to mention that, but when we were looking at the cultivar evaluation material in this past year, we had Raider that was treated and Raider that was not treated. And there was a difference in there. Non-treated was 8 and treated was 4. So that's 50%. So that's kind of a teaser. That's something that we need to look at, that particular product and maybe other seed treatment applications as well, as a way of delivering these products. And transplants. I put that in there because perhaps one thing we could do is grow plants in the greenhouse, treat them with something so that when they go into the field, they're treated already. Perhaps that'll give that plant a leg up on surviving. And perhaps doing that in conjunction with perhaps a resistant crisphead cultivar that we have already, and combining those two. Perhaps that will work. Again, we're open for any suggestions, and if you have any, please contact me. There's my cell phone and email address. And that's pretty much what I have to say. Yes?
Yeah, I was talking to a grower who indicated that the seed tape that Brian Antell has developed, that was at the summit, there's going to be about 4,000 acres put in. And I was just wondering, since you said the disease attacked at the radical as soon as it started, whether you're planting a live plant, that material is biodegradable, whether it could be impregnated with a chemical when that little seed tape is planted, whether that would help.
Right. Yeah, that's actually what we're thinking of looking at in cooperation with that grower too. Because even just getting the plant in there, even if you did not treat it, the plant at least has a start. And then if you could impose on that a seed treatment, or not, it'd be a plant treatment at this time, perhaps it will help in conjunction with some of the genetic resistance that's in the crisphead variety already. We can get to a marketable head, which is the ultimate goal. Any other questions? Yes, sir.
Is there any race determined by differential variety? And if so, would it be interesting to get some of the Japanese varieties and test them along with the material? What are they doing with themselves?
Well, that's a multiple question. Race typing is done by using differential varieties, but there are other methods as well, and Barry could address that more intelligently than I could. So it is possible to look at whether you have race 1, 2, or maybe a new one by not using the plant, DNA methods, and Barry can actually do that. And so that is something that we can use going forward. Any other questions? Any questions on solarization that weren't?
Right, question on
Yep.
solarization in that. What's the cost of that plastic per acre? Do you have an idea?
I don't. We did that, obviously those trials were done at least 10 years ago. The plastic itself, you can use the thinnest, it doesn't have to be highly expensive plastic. So the cost basically is laying the plastic and then disposing of it later.
Do you remember what the mill strength was on it?
I believe it was half a mil, I believe. I'd have to check that.
Can the plastic be reused?
No. No, it's pretty thin, and in the process of, now everything that we did in laying plastic was all manual. If you were going to do this commercially, of course, there are machines and implements that'll lay the plastic as well as remove it. But this was all kind of, using shovels, and one of the problems we have with keeping plastic in place, of course, is the wind that we get during July and August. And so you have to be very careful if you do this that the plastic is anchored in the soil so it stays where it'll be doing some good. All right? Okay, well, thank you very much.
Well, let's thank our 2 researchers. And I want to thank all of you guys, because we couldn't do this without your guys' input into what's working, what's not, what your ideas are. We brought a bunch of growers in recently to talk about year 3 and year 4 trials. What should we do? Mike mentioned seed coating, the injection came up. We want to facilitate all of that so that if just spraying over the top is not the ideal method for some of these products, that we do it in a way that they think will be, have the most efficacy. So we're going to try cultural practices, but I'm kind of the village idiot here. We need your guys' input to tell us what we ought to be doing. So we appreciate that. We appreciate the support you've given. Certainly appreciate these guys for working with us. And I think I don't know whether I can say we've made advancements, but we're certainly advancing as far as knowledge and things that we're looking at. So I feel good that we're making some headway. I do want to recognize, Matt left, but JV Farms, TNA, and Gila Valley Farms, thank you, Larry Ott, provided the fields for us to do this in, and that's been super helpful. The seed companies have been really excited about, having an actual field trial to do them in, so that's been great. But yeah, any feedback that you guys have that can help us to try something, see if it works, we're certainly open to that. So I think we'll wrap up. These were on your seats. There's a lot of information on our website, and we're continually adding to it as we get trial results, presentations, all that stuff. I do want to recognize Rosa Bevington, who's working with us now, and we're doing this YouTube live, and it'll be on our website and on YouTube if you want to share this with anybody. And Sonnet Nelson's out back, but Sonnet's the other member of our team that really made this happen. So thank you guys for coming, and we'll keep working.